This conference will now be recorded. This conference will now be recorded.
Ma'am, you are not audible, ma'am.
This
This is
conference will
following
now be recorded.
microbiology. Okay, I am the part-time lecturer in Biosetup Life Sciences. Okay, I am also doing as a research in microbiology, and I am working as an assistant professor in biotechnology and microbiology in Kavito Degree College, Kakatiya University, Warangal. Okay, I have qualified GATE and also TSET, and these are all my information about my information. And today I am going to discuss about the staining techniques. Staining techniques. I think all of you are familiar with this topic because in degree and BSc level and PG level, I think all of you know about this. Okay, what is general staining? Staining means why we are doing staining. Okay, actually, these staining techniques, these are mainly observed in microbiological techniques to observe the different pathological microorganisms. And why these stainings are used? For the differentiation of pathological microorganisms or the pathogenic microorganisms. And that means here mainly these techniques are mainly used to study the microscopic microorganisms. Microscopic organisms. Actually, these microscopic organisms, these are colorless ones. We have to observe these colorless microorganisms present in the particular sample means first these are to be stained with different types of stains or dyes, and that causes the visualization of different pathological microorganisms. Here, actually, in microbiology, we study the culturing techniques, isolation techniques, and here, for the isolation and culturing means, first we have to identify the particular species present in that one, in that particular sample, and that means the particular organisms present in the cultural samples are identified by mainly staining techniques or by using the different dyes. Okay, here these dyes, and that causes the enhancing of contrast, enhancing of contrast in the structure or in the shape, and that causes the visualization of particular microorganisms. That means the staining techniques are mainly used to visualize the structural ones, shape, and other arrangements that are present in the particular microorganism. Here normally microscopes are mainly used for the visualization of microorganisms. And that means here to to observe under the microscope means we have to enhance the contrast and that are present in the sample, and that enhancement of contrast is mainly done by using the stains or dyes. Stains or dyes are mainly used for the visualization of microscopic organisms. And in the microscopic organisms, different microorganisms have different structural ones. Structural arrangements are different and shapes are different. And based on these differences, different types of stains are present. Different types of stains are present. For example, for the normal observation of shape of the microorganisms, any one of the stains are used for the enhancement of contrast to observe them under the microscope. And that means depending upon the characteristics to study, here different types of stains are present. And that means stains are mainly used for the enhancement of contrast to visualize under the microscope. And here mainly stains are used for the visualization purpose. And stain is a dye of color and that causes the observation of microorganisms under the microscope. And by using these living microorganisms or dead non-living microorganisms, and these can be observed. And also for the studying of histopathological examinations, that means for the tissue structural studies also, different stains are present. And that means for the studying of structural arrangements and for the shapes, and these staining techniques are mainly needed. Okay, and that is a basic introduction to staining techniques. Here, mainly microscopic organisms. For the visibility of microorganisms, these are used. And here different types of staining techniques are present. Mainly simple stain, differential stains. And that means to differentiate the different groups of microorganisms. And that includes the gram staining processes, Csir Nielsen staining techniques, and special arrangements present in the structures. And that can be studied by using the special stains. And that means staining flagella structures, capsular structures, endospore structures, and these are studied by using the staining techniques. First, we have to understand the staining techniques means first the structure of bacteria should be considered. And what is the structural components present in bacteria? Anyone chat with me? Normally, the structural components present in bacteria. Structural components, that means the basic structure of bacteria contains which parts? Okay, cell wall, endospore, flagella. Yeah, good, ma. Okay, here mainly the first structural part is the cell wall, and that is the cell wall is the main structural part. And next plasma membrane, and next structural arrangement that contains the flagella, capsule, and these are the structural components. And inside the bacterial cell, plasmids are present, ribosomes are present, and that means the nuclear material that is present, and that means these are the structural components. That means here For studying for visualization of bacteria means, and these structural components can be studied and that are understood by using the staining techniques only. Staining techniques are only used for the identification of different structural arrangements, and that is called as the staining. Here, stains are used for the observation, for the visualization. That means different types of stains are present. Here mainly acidic stains, basic stains, neutral stains, and these are mainly present for visualization of any type of microorganisms. Acidic means these contain negatively charged acid radicals. For example, acid means acetic acid, and that means it is in the form of CH3COOH. When it undergoes ionization form means, and that gives rise to the CH3COO minus plus H plus, and that means it giving the acid radicals that are in negatively charged, and that negatively charged acid radicals, these are mainly used to stain basic components of cell structure. These are mainly used to stain basic components of cell structure. The basic components of cell structural means the cytoplasmic portion. The cytoplasmic portion structure, and that is mainly basic component. And that can be studied by using the acidic dyes. Here acidic dyes means anionic ones. That means the negatively charged ones, and that gives the colors, when staining the that staining the basic components, and that are in the cytoplasm, and that are called as acidic dyes. And for these acidic dyes, main examples and that are of eosin, acid fuchsin, malachite green, nitrosin, Indian ink, and these picric acid, and these are all main acidic dyes. Acidic dyes are the anionic ones, and that are mainly used to stain basic components of cell. Okay, and next coming to the basic dyes. And basic dyes means here these are the cationic ones. Cationic, which charge is present? Cation means which charge is present? Cations means positive charge. Good, Vidishi. Here, that means the cations means the positively charged radicals, and that combines with the negatively charged particles. And that means the negatively charged ones are mainly that are present in bacterial structure or nucleic acids. That means the basic dyes, they mainly interact with the nucleic acid structure. That means the basic dyes are the positively charged radicals, and that combines with the negatively charged particles in the cytoplasm and gives color. And here the main basic dyes, and that are of mainly hematoxylene, methylene blue, crystal violet, gentian violet, safranine, and these are the some of the examples, and that are included in basic. Here acidic and basic. And next coming to the neutral ones. Neutral means neutral charge. That means both positively and negatively charged ones, they impart different colors to different components. That means they giving the colors by giving different charges. That means positively charged ones are present, negatively charged ones are present in that particular dye, and that gives different colors to different components. And for this mainly example is the Ginseng stains and Leishman stains, bright stains. These are the different types of stains for the observation, for the visualization of different pathogenic microorganisms. And these are the different types of dyes. Okay, and what are the staining methods? And that are present for the visualization of microorganisms. Here, positive staining is present, negative staining is present. Mainly, the positive staining. What is meant by positive staining? Here, actually, the cells are themselves colored and also appearing in clear background. That means the colored cells are directly absorbed by background, by clear background. And that technique is called as the positive staining. Positive staining. And here, in the positive staining, different types of staining techniques are present. And that has simple staining, differential staining. And what is meant by simple staining? Here, simple means just simple dyes can be used for the visualization. And that means here is green, and that gives the color contrast and gives color to the all bacteria or cells that are present in the cultural samples. That means, for example, if the culture contains E. coli or any other microorganisms, and that means a simple stain is used for the visualization, that giving the color contrast, and that technique is called as the simple staining technique. And for these simple staining techniques, different types of dyes can be used. And for this low flush methylene blue, polychrome methylene blue, carbolfuchsine, and saffronin, and crystal violet, and these can be used for the simple identification of particular microorganisms present in the cultural sample. And that type of technique is called as simple staining technique. And second one, differential staining. Differential, the meaning also there. Differential means different colors to different pathogenic microorganisms, to different microorganisms. And that means the stain which imparts different colors to different microorganisms. And that is called as differential staining. Here, that means here more than one stain is used for the visualization of microorganisms present in the cultural sample. And that methods are of mainly gram staining, acid fast staining, special staining. Gram staining is mainly used to differentiate gram positive ones and gram negative ones. Whereas acid fast staining technique, and that is mainly used for the identification of mycobacterium species. Mycobacterium tuberculosis is there, Mycobacterium leprae is there, and to identify these ones, this acid fast staining is used. And also special staining techniques are also present. Special means the structural arrangements, and that can be identified by using these differential staining techniques, and that comes under the positive staining techniques. And next, coming to the negative staining technique. Here, in the negative staining technique, the cells remain uncolored. Cells are in uncolored and the background is colored. That means the colored background is mainly used for the visualization of colored microorganisms. Uncolored microorganism. And that is called as a negative staining. Here the cells are in uncolored, background is colored, and that means here for the better visualization of different images, and that means different pathogenic microorganisms present in that present in that culture. And here the negative staining technique is used. Mainly negative stains are of Indian ink, Negrosin, and these are all negative stains. And these are the types of stainings: positive staining and negative staining. And that means here first we have to do the staining, means first we have to prepare the smear, bacterial smear. And what is meant by smear? Here it is the culture, bacterial culture. That means the bacterial culture, it is mainly is distributed as the bacterial cells on his slide for the studying them under the microscope. Any microscope can be used depending upon the magnification. Okay, here mainly smear is to be prepared for the visualization of microorganisms under the microscope. And how the smear is prepared? And that means here first normally the culture is spread over a slide surface. The culture is taken and it is normally spread over the slide surface. And then in the next step it is allowed for air dry. Allowed for air dry. And then it is heat fixed. Heat fixing occurs. That means heat fixing means for the cells to adhere to the solid surface. We are using the slides. That means for to adhere to the solid surface, the microbial cells, these to be done by the process called as heat fixation process. Heat fixation. And that means for the smear preparation different steps are present. First take the small sample of culture, and next this culture is to be air dried on the slide, and then it is to be heat fixed. Sometimes some fixation methods are also present for the preparation of smear. See the slides here, just we are taking normal slide, and in that the culture of sample is placed, and here only that sample, the culture, the small culture is spread over the slide, and that when it is air dried and heat fixed, that causes the smear, and that is called as the smear preparation. Smear preparation. It is mainly for the observation of microorganisms. Actually, for the histopathological examinations, also the smear is to be prepared. Here, actually, the stored samples, how can these to be used means, actually, the culture samples are mainly preserved by different methods, by applying paraffin, by using the soil, and different methods are there for the preservation of microbial cultures. Here, when the preserved cultures are to be taken means, and that means here that layer is to be removed. For example, paraffin layer is present, and that to be removed for the smear preparation. Here we have to add the stains. That means to take up the stains, and that means the paraffin layer is to be removed. Here, the paraffin layer is first removed with the xylene, and then xylene is removed with the alcohol, and this alcohol can be replaced with the water. That means for the clear visualization of preserved sample or the tissues, and that already layered with the paraffin. That means here first the paraffin layer is to be removed, and then these to be prepared for the smear, and these to be taken for the visualization process, and that for the mainly tissue sections. Here smear fixation can be done by using the heat fixation or by using the methanol fixation. Normal heat fixation means just we pass the slides through the flame 2 or 3 times, and that means the passing of the slide onto the flame 2 or 3 times, that causes the heat fixation. And before this staining, and that slide should be cool. That slide should be good. And that is the normal smear fixation for any microorganism fixation. And also methanol fixation is also present. Here, the methanol fixation, for the methanol fixation, the air-dried smears, these are to be placed in the jars and that containing methanol for 1 minute. And also, the slide should be flooded with the methanol for 1 minute, and that causes the staining of that particular microorganisms present in the culture. And this is the way for the observation under the microscope. General staining process includes the preparation of smear and then addition of stain and then observation under the microscope. And these are the three steps that are included in staining process. First step, what is the first step? Preparation of smear. Second step is the addition of stain. Third step is the observe under the microscope. These are the three steps that are included for the staining technique. And here, for this fixation is to be done. And next, coming to the different process of stains. For the observation of different shapes, for the structural arrangements, and that can be done by using the different staining techniques. First, coming to the simple staining techniques. Simple staining, that means single stain, is used for the visualization of microorganisms. And here, not only the microorganisms, different types of cells that are present can be also visualized by using the simple staining technique. And for this, mainly methylene blue, that called low flush methylene blue. It is the most useful stain, and that mainly gives the characteristic morphology of different types of cells. And here, different morphological structures of microorganisms can be studied by using the methylene blue stain. Here, polymorphs, lymphocytic cells, and other cells, and that are clearly visible by using the simple stains. And here, normal the simple stains may be crystal violet, saffrony, carboxyl, or any other type of stains can be. Okay, here and next, polychrome methylene blue. Polychrome methylene blue means it is the modified form of methylene blue. This is a modified form of low flush methylene blue. How it is made means this is made by allowing low flush methylene blue to ripen slowly. That means that causes the variation, the changes that occurring in methylene blue. That causes the polychrome methylene blue. Here, the slow oxidation of methylene blue in low flush methylene that forms a violet compound, and that causes the staining of different polychrome properties. That means different colored ones can be observed by using the polychrome methylene blue, and different structural arrangements can be visualized by using the polychrome methylene blue. And next, here during the ripening, oxidation is taking place, and to this also 1% potassium carbonate should also be added for the ripening process. For the complete ripening means it should be added by using the 1% potassium carbonate, and that gives for the polychrome reactions, that causes the polychrome reactions. Actually, the methylene blue, it normally appears, it causes the blue staining, whereas in polychrome, this one polymethylene blue, here the structures are observed in the form of red purple color. Red purple color. Here, for this main example is acid capsular material of Anthrax bacillus, and that can be studied by using this methylene blue. And this is the mainly dye used for the identification of acid capsular structures in anthrax bacillus. Okay, and other ones can also be used for the simple staining methods, and that may be dilute carbol Here, carbol it is made by here dilute ones we are using. That means the dilute carbol mainly it is made by diluting the Gill-Nielsen stain with with 10 to 20 times its volume of water. That means by slowly diluting the normal stains also here, and that are mainly used for the simple staining process. Here, when that means the concentrated ones are used means that causes the blurring of the image, and that means to for for the clear observation of the sample means we have to dilute that particular stains, and that causes the simple staining for the visualization of different structural ones. Okay, and here normal the requirements for the simple staining or the stains that are used for the simple staining that are of low flux methylene blue, carbolfoxin, crystal violet, safranin, and these can be used for the visualization of simple structural arrangements present in the microbial parts. And here, first the requirement for the staining technique is the smear preparation. That is the fixed smear. And to observe these staining means compound microscope can be used. Okay, and sometimes here we have to enhance the magnification, and that magnification is enhanced by using the oil. Oil is mainly used. Okay, what is the procedure involved in simple staining? First, in the simple staining, what is happening? First, the clear smear. Smear is prepared on the slide, and then heat fixing of smear, and that by passing the slide 2 to 3 times on Bunsen burner plate. And next, after cooling the slide, just the smear is allowed for the stain, and this is allowed for 3 minutes, and that causes a clear observation of the stain. And then here the stain is washed, that means it is absorbing or not, and that is clearly absorbed by washing the slate with water, and then it is air dry, and then this smear it is absorbed under low power object and also oil immersion object. Here, the magnification is enhanced by using the objectives. 10x, 40x is there, 100x are there, and that means objectives are present in the compound microscope. And that means here the presence of microorganisms and the cellular contents that are present in the sample are observed mainly by using the microscope, and this is the process that is generally involved in simple staining process. Okay, here when the cells are stained by using the low flux methylene blue means here the cells are appeared in this form, and that means the blue colored cellular structures, and that can be observed by using the simple staining technique. Okay, and next coming to the differential staining. In differential staining, mainly it is used for the differentiation of microorganisms. That means differentiation of the particular individuals that are present in the cultural samples. Here, gram staining mainly differentiates the bacteria into two groups. And here, based on the structural components only, these two groups are differentiated. And that causes the gram positive ones and gram negative ones. Gram positive bacteria is there and gram negative bacteria is there. And here, and these can be differentiated by using the gram staining technique. And what is the main principle involved in gram staining is here the Gram's reaction it is mainly depends on the permeability of bacterial cell wall and cytoplasmic membrane to the dyes. Here, different types of dyes are used. More than two dyes. Differential stains means more than two dyes. Two dyes are mainly used for the differentiation. Here, here the Gram's reaction, it is mainly depend on permeability of the bacterial cell wall and cytoplasmic membrane structure. That means these structures, these are allowing the staining solutions to enter into the cell, and by taking up these dyes only, the structures can be visualized. That means the permeability character of that bacterial cell wall and cytoplasmic membrane, and that causes the differentiation of gram gram stain. That means differentiation of bacterial species into gram positive ones and gram negative ones. And here mainly the stains used here mainly are of crystal violet, grams iodine and safranin. Mainly two types of stains are used here. Primary stain and secondary stain. Primary stain, that means that is the first used stain. That is called as the primary stain. And that causes the taking of the dye first. Okay, next, second dye is used, and that called as the counter stain. And in between these also, grams iodine is used. Grams iodine is used, and that means that acting as the mordant. Here, what is the main principle in observing these gram positive and gram negative bacteria means here the dyes that causes the visualization of bacteria by taking up. Here, for example, gram positive ones are there, gram negative ones are. Gram positive ones taking up of one particular dye, whereas gram negative ones are also taking up the dye. But here differentiation is present by taking out different dyes. And that means differentiation is based on the taking up of the dyes. Here, the crystal violet iodine complex, and that combines with the bacterial molecule, and that causes the precipitation within the cells. Precipitation within the cells. Here, alcohol is also used during the staining process, and that alcohol causes the decolorization or dehydration of the cell wall. That means not only the cell wall structure in bacteria, and that is mainly made up of with the component called as peptidoglycan. Peptidoglycan. And what is meant by peptidoglycan? Anyone? Peptidoglycan. And that containing which portions? Peptidoglycan means proteins and carbohydrates. Okay, polymer of NAM and NAG. Okay, good ma. And that means here NAM and NAG are the carbohydrates, that are the glycan portions, whereas the proteins that are present here are peptide bonds that are linked by peptide bonds. What are the amino acids that are present in peptide portion? Amino acids present in peptidoglycan. What are the amino acids present in peptidoglycan structure? Okay, lysine, good. Orchidine, okay. Adenine, okay. Here, the peptidoglycan portion, right? Good mark. Here, mainly it consists of the glycan portion and peptide portion. And the glycan portion mainly consists of the NAG and NAM. And that are mainly linked by beta 1,4 linkage. And it consists of alternating amino acids, alanine, glutamic acids. Okay, and here in place of glutamine, sometimes lysine is also present. Not valine, valine it is not present here in peptidoglycan. Priyama Kumari, valine is not present here. Sorry. Okay, here in gram positive bacteria, what is happening means the structural component of gram positive bacteria mainly consists of 80% of peptidoglycan portion. 80% of peptidoglycan is present. That means more amount of peptidoglycan is present. And here we are using different dyes for the visualization. That means the peptidoglycanic portion, and that causes the uptaking of these dyes. Here, when the alcohol mixture that is used as the decolorizing agent, and that causes the decreasing of the space between the molecules in between the cell walls, and that causes a trapping of crystal violet only. That causes the crystal violet iodine complex trapping into the spaces also. And that means here the gram positive bacteria, they do not get decolorized with the alcoholic solution, and they only retain the primary dyes, and that are appearing as the violet color, appearing as the violet. Okay, and here the gram positive bacteria, these contain mainly acidic protoplasm. Hence, these bind to the basic dyes more frequently. That means the basic dyes are mainly used for the visualization of gram positive. Already I said what are the basic dyes, what are the acidic dyes. Okay, and here, and this is the gram positive bacteria. And next, coming to the gram negative bacteria, what is happening in gram negative bacteria? The peptidoglycan portion is less. The peptidoglycan portion is less, whereas it contains the lipid component more. The lipid component is more in gram negative bacteria, and that causes the here the primary dyes which are used in, which are primary stains which are used for the gram stain, and that are not, that are decolorized by using the decolorizing agent, and hence it takes up the counter stain at the secondary state, and that causes the, that appearing as the secondary counter stain color. That means, for example, in the first primary stain, crystal violet is used. In the counter stain, saffronin is used. Crystal violet means, and that causes the appearance in the form of violet color, whereas saffronin means pink colored ones. Here, the crystal violet color absorbed means that causes the gram positive ones. Whereas in gram negative ones, these are appeared as pink colored ones. Pink colored colonies are appeared. That means these are taking up the counter state. And that is the main principle here. And coming to the structure, this is a normal basic structure, that is a gram negative bacteria, and that contains the mainly phospholipids, bacterial lipopolysaccharides outside of the peptidoglycan layer. Okay, and it also contains the in between outer membrane and peptidoglycan periplasmic space is present. Okay, and this is the normal structure of gram negative bacteria. And this is the differentiation between the gram positive and gram negative. Gram positive, that mainly consists of the cell wall portion, and that is the peptidoglycan portion, whereas the cell membrane is the less portion. And here, whereas coming to the gram negative ones means here the cell wall that contains the thin region of peptidoglycan. Whereas the lipid region, it is occupied by more amount of the lipid region. And that means the outer membrane is present and the inner cell membrane is present. And the outer membrane is mainly rich with the lipid region. And that is the main difference between the gram positive and gram negative. And also another difference is that echoic acids and echoic acids are mainly present in the gram positive bacteria. And that is the basic difference in the structure of gram positive and gram negative. And based on this, here gram staining is mainly used for the differentiation of gram positive and gram negative. Here, what are the steps that are mainly involved in the staining? Here, first, it should be first stained with the primary stain, that is the crystal violet stain. First, as usual, the process is similar to that of simple stain. First is the preparation of smear, and next the smear is after the preparation of smear, it is to be stained with the crystal violet, and then it is to be stained. Then it is to be added with the iodine that acts as the mordant. That means here that forms the crystal violet iodine complex. That means in gram positive bacteria, mainly this complex causes the violet color colonies. And next, decolorizing agent, and that is alcohol is added. And that causes the decolorization of primary stain. Decolorization of primary stain. And that means here when the cells are decolorized with the alcohol mix, and that causes the gram negative ones. If the decolorization is not possible, decolorization is not possible means, and that appeared as a violet color. That means here gram positive cells remain purple, whereas the gram negative cells that become colorless. And next here colorless ones, and these are again stained with the counter stain. That means another stain, another dye is used for the staining, and that causes the observation of cells in the form of pink color or in red color when these are counteracted with the different stain. That is the saffron is mainly used as the counter stain. And this is the main staining steps. These are the main staining steps that are involved in the visualization. Mainly here primary staining, and here mordant is also to be included here. Mordant, decolorizer, counter stain, and these are to be added. Mainly primary stain crystal violet, here mordant gram iodine, decolorizer, alcohol acetone mixture, or both can be used for this decolorization. Next, next counter stain, that is the secondary stain, and here the secondary stain is the carbol. Okay, and this is the main staining method for the differential staining. Here, primary staining, and next mordanting, decolorization, counter staining, and that causes the visualization under microscope. And if the magnification is to be increased means this ought to be observed under the oil immersion objective. Okay, and here what is the result means? And here that gives the purple colored ones and pink colored ones. And that means here gram positive ones are observed as the violet colored colors, whereas the gram negative ones means these are observed as the pink colored colors. And this is the result, and that gives the gram string. Okay, and also for the tissue sections can also be done the same technique. Here, tissue section means here like the fibrils, keratohyalins, keratins, and these structures can be visualized by using the gram staining technique. Gram staining technique, and that means here the some concentration of stains should be increased for the staining by using the staining for the tissues. Okay, and here the gram positive organs that are present in tissue section and that are absorbed in the form of purple color. Whereas the gram negative organisms, these are appeared in red color. Okay, here the same technique. First crystal violet, tap water, rinse with the tap water, then iodine solution, then with the decolorizing agent, then counter stain with the neutral ring. And that causes the visualization of gram positive and gram negative. And also, and this is a normal technique, just normal basic easy technique, quick gram staining technique. That is the first add with the primary stain for about 5 seconds, and next iodine solution for about 5 seconds, and next this is to be washed with the water, then decolorizing agent for 2 seconds, then it is to be washed, and then next counter stain is to be added, and that gives the observation. Okay, here this gram staining this technique is modified by different scientists, and that causes that means here the stains that are used in gram staining are modified by different scientists, and that uses the Koplow-Biermann's modification, and here he used the primary stain methyl violet decolorizer as the acetone alcohol mixture. And Jensen used the primary stain as the methyl violet. Decolorizer is the absolute alcohol, counter stain is the neutral. And next, Preston and Morales. Here, primary stain is crystal violet, decolorizer is the iodine acetone. Next, Veget's he used the primary stain as the carbol gentian violet. And decolorizer is the aniline soda, and that means it is mainly used for the tissue section. That means the modifications are done by changing the stains only. And what are sometimes the stain during the staining process, some mistakes are done, and that causes the errors in staining. And what are the errors that are observed? That means excessive heat. Excessive heat causes the just complete degradation of microorganisms. That killing of microorganisms occurs by the excessive, and also concentration of stain is also to be maintained, and washing also clears all the microorganisms present in the culture. Iodine exposure, that means the particular time intervals should be maintained during the addition of different states, different reagents. Okay, and these to be maintained during the process, and if these the conditions are not maintained means, and that causes the errors in staining process. Okay, and what are the main applications of this grain staining? What are the main applications here? Here, this is mainly used for the diagnosis of diseases such as mainly bacterial meningitis or other bacterial diseases. That can be for the identification of Escherichia coli organism. These can be done by using the gram staining, and also antibiotics for the selection of different antibiotics. That means here, when the pathogenic microorganisms only identified means, that causes the giving of particular antibiotic. That is the selection of antibiotics that depends on the particular pathogenic microorganism, and that means the finding is depending upon the staining procedure only. Okay, media we have to use subculturing process is to be done, and that means the staining procedure first it is to be done for the identification of particular microorganisms. Okay, and also for the screening of different clinical samples, and that containing different types of tissue cells, pus cells, epithelial cells, and these are screened by using the gram stain. And also counting of bacteria, morphology, and different types of bacteria that are present in clinical specimens, and these are identified by using the gram staining techniques. Okay, and this is the main introduction to the staining techniques, simple staining, and in differential staining, mainly the gram staining is used for the differentiation of two bacterial species. Okay, Understood, ma? Any doubts regarding this?
Dear students, if you have any doubts regarding today's topic, just ask your doubts in the comment section.
Okay, and this is the main topic that is related to the staining, and in the stain different types of questions that are arising during the CSI net exam or IIT JAM exam. Okay, any doubts regarding this? You can chat. Okay, ma. Okay. Okay, some of the questions. I will discuss some of the questions here. What is an example of cationic dye here in this? Malachite green, eosine, picric acid, all. What is the answer for this? Example of cationic dye. C. What are cationic dyes? Cationic means basic or acidic dyes. Basic dyes. Basic dyes that are mainly used to stain acidic components like nucleic acid. And that means what are the basic dyes? What are the basic dyes? Example for basic dyes. Yeah, methylene blue, crystal violet, saffronin. These are the basic dyes. Here, eosine picric acid. These comes under the acidic dyes. Eosine picric acid. These are the acidic dyes. That are the anionic dyes. Whereas malachite green, and that is the basic dye. That is the cationic dye. That means option A is the correct one. Option A is the correct one. Example of cationic dye is the malachite green. Understood, ma'am? And next, simple staining is used for. Simple staining. Simple, just simple. That means studying cellular organisms. Yeah. Good. Dikshika Datta, good ma. Vidushi, good. Just basic, that means revealing basic cell shape. That is the correct answer. Second option is the correct one. Priyama Kumari, good ma. Okay. Next, the third one. Gram staining is mainly based on which? Option A, option B, option C, D. Which one is correct? Option C. Okay. Option A, no. Observe this once. Here, gram staining. Gram staining is mainly based on which? Here, amount of lipid present in cell wall. This is the structural component. Already I am said what is the principle behind in gram staining. Here, Lipid present in cell wall means that is only present in gram negative ones, and that means it is not differentiating. And next, coming to the third option, amount of peptidoglycan present in cell wall. Here, the peptidoglycan is not responsible for taking up the dyes. It is not responsible for taking up the dyes. That means here high amount means it takes up the dye, but it does not depending upon the peptidoglycan portion. It depends only on the porosity of cell membrane and cell wall. How much amount of the dye is passing through the solution, is passing through the cell structure. Or it depends on the how the decolorization occurs. How much amount of decolorization? That means here the stains which are used in gram staining, and that causes the porosity and that permeability of cell membrane and cell wall. Okay, here the permeability of cell membrane and cell wall, and that combines with the porosity of cell membrane. Okay, and also it depends on the peptidoglycan. Here, option C is the correct one. Actually, the answer is the permeability of cell membrane and cell wall. Actually, the answer is permeability of cell wall and cell membrane. Here in the answer, amount of peptidoglycan is correct one. Okay, Shalini, okay ma? Dipshika, good. Amount of peptidoglycan present. Actually, the correct answer is the permeability only. Okay, next, gram staining is also called option B. Good. Good, Dipshika, Shalini, Vidishi. Differential staining, because it causes the differentiation of two bacterial species, gram positive ones and gram negative ones, and that is called as the differential staining. And the fifth one, what are the steps for gram staining? Correct step. Addition of crystal violet. First step is the addition of crystal violet. Third, next one is the addition of mordant. Sorry, here the correct answer. Here first step is the addition of crystal violet, then with the mordant, and then with the decolorization with the alcohol, and then with the addition of safranin. First one is the 2, next 1, 4, 3. Here option 2, 1, 4 and 3. And this is C option is the correct one. Observe these steps. First we add the primary stain, that is the addition of crystal violet. Second one is the addition of mordant, and that is the iodine that causes the crystal violet iodine complex. Then decolorization with alcohol. And that means 2, 1, 4, 3. Okay, next counter stain. Okay, here C option is the, and these are the some of the questions and that come across in these staining techniques. Okay, any doubts regarding this? You can ask me.
Okay, ma'am. So, students, if you have understood today's topic, you can just comment yes in the comment section. Okay, okay. So that's all for today. And those who will be missing this lecture, they can watch in our YouTube channel. And once again, I would like to repeat, 10th November is the deadline. Please make your registration before 10th November. After After 10 November, there will be no demo classes. So please, first 30 comers will get concession of 500 rupees, and you can pay in the installment also. First term you can pay 4,000, and the next term you can pay 3,500. Hurry up, only 8 days left. So Argdeep, now you can put the